Class, when you open a bottle of spoiled yogurt and are greeted by a pungent sour odor, have you ever wondered, why does homemade yogurt often fail while industrial production succeeds with stable output on the scale of tens of thousands of tons using the same milk and lactic acid bacteria? When news reports surface about pharmaceutical factories losing millions due to contaminated batches, have you considered how a single foreign microorganism infiltrating the fermenter can disrupt an entire production line? From the white film, calm yeast, crisis in rustic sauce vats to the phage panic in antibiotic workshops, the term contamination hangs like a sword over the fermentation industry. Today, we will confront this survival battle in the microscopic world head-on. Let's study the topic of pollution control in biological fermentation processes together. Our lesson today will focus on 4 primary areas. 1. The impact of contamination on fermentation. 2. Analysis of the causes of pollution and prevention and control of contamination during biological fermentation. 3. Inspection of pollution in biological fermentation. 4. Prevention and control of phages and emergency measures in the event of pollution. The effects of contamination primarily hinge on the types of polluting microorganisms and the timing of their intrusion. Let us first understand the impact of contamination on fermentation. Firstly, contamination by different microorganisms with varying characteristics brings different risks. Phages are highly infectious, spread rapidly, and are difficult to control. Once contamination occurs, output drops sharply, and in severe cases, entire batches must be discarded, leading to cessation of production. Certain unwanted bacteria trigger autolysis of production strains, generate excessive foam, and interfere with aeration and mixing, while others cause the fermentation broth to develop unpleasant odors, become sour, and drop in pH, thereby damaging acid-sensitive products. Additionally, Bacillus species form heat-resistant spores that are difficult to eradicate and, once present, tend to cause repeated contamination. Secondly, the timing of contamination affects outcomes differently. Contamination at the seed culture stage significantly reduces seed quality and requires sterilization, discarding of the batch, and thorough inspection of equipment and pipelines. Early-stage contamination leads to competition for nutrients between unwanted organisms and production strains, which can be addressed by sterilizing, supplementing the substrate, and reinoculation. Mid-stage contamination is the most detrimental, severely disrupting cell growth and product formation and thus must be tackled promptly according to the specific circumstances. In the late stage of fermentation, accumulated products offer some inhibition against contaminants. If contamination is minor, fermentation may proceed, but if it is significant, early termination of the batch is advised. Now let's look at the causes of pollution and the prevention and control measures in the biological fermentation process. These mainly include seed contamination, which requires rigorous inspection to prevent the introduction of unwanted microorganisms, airborne contamination, which must be countered with high-efficiency filtration and similar measures, equipment leakage, which needs to be averted through regular maintenance and repair, incomplete sterilization of media, which can be rectified by optimizing sterilization procedures, and inadequate technical management, which should be remedied through improved technical oversight and standardized operating procedures. Collectively, these measures constitute the key links in the prevention and control of pollution during fermentation. Next, let's examine pollution inspection in biological fermentation. Main methods include microscopic examination, often using gram staining to characterize and distinguish production strains from contaminants, supplemented when necessary by spore or flagella staining, which remains the simplest and most direct approach. Streak plating or slant cultivation methods, whereby pre-prepared plates are incubated at 37 degrees Celsius for 24 hours to confirm sterility before samples are streaked, then incubated at both 37 degrees Celsius and 27 degrees Celsius. Observations at 8 hours reveal contamination, and if suspicious colonies are detected on 3 consecutive occasions, contamination is confirmed. Furthermore, abnormal phenomena during fermentation, such as atypical cell growth, reduction in cell count under the microscope, abnormal optical density, OD, irregular sugar consumption, decreased product synthesis, changes in pH, or excessive foaming, can also serve as indicators for contamination. Finally, let's discuss emergency measures following phage contamination. This includes addition of small amounts of chemicals, such as oxalic acid or citric acid chelating agents, to inhibit phage adsorption and reproduction, using certain antibiotics to suppress phage protein synthesis and proliferation. Though this approach only works with antibiotic-resistant production strains and, due to high costs, is not viable for large-scale fermenters. In the event of phage contamination during fermentation, fresh culture media or growth factors can be supplemented to promote the growth of production strains, accelerate the fermentation process, and enable successful continuation of fermentation. Large amounts of seed broth or cultures of resistant strains can be inoculated to carry fermentation through to completion and minimize losses, thus preventing batch disposal. Prior to re-inoculation, infected fermenter broth may also undergo low-temperature sterilization. Class, through today's lesson, we have gained an in-depth understanding of the key technologies and methods for pollution control in biological fermentation. This knowledge not only reflects the rigorous scientific attitude demanded in bioengineering, but also demonstrates China's technical capabilities in fermentation industry development. As food fermentation professionals in the new era, we must always remember the industry's guiding principle: quality is life. We should approach every stage of production with craftsmanship and an unwavering commitment to excellence. Let us be guided by scientific innovation and assume our social responsibility, dedicating our energies to advancing the high-quality development of China's biomanufacturing industry, and fulfilling our mission to serve the nation through science and technology. This concludes today's lecture. After class, you are required to summarize and respond to the following 2 questions. What is the most appropriate response to mid-stage contamination in fermentation? How should phage contamination be addressed? That's all for today's class. Thank you all for listening.